activities. And part of that mandate is to develop this long-term time series that measures a variety of different ecosystem indicators developed from this project. The New York Bight is a very diverse ecosystem. There are hundreds of marine fish species, a variety of different charismatic megafauna like whales, migrating seabirds, shark species, many different types of zooplankton, all which have commercial and ecological importance. And there's been a few talks, like some plenary talks, and a few posters, and a few more talks that will be happening later about offshore wind. On the map here is a BOA map of the improved wind energy areas in the shaded green region. So there's planned development of the New York Bight, so it's really important to have, again, a baseline data set to look to really be able to evaluate the impact of these activities and development. So our survey comprises 7 cross-shelf transects going from coastal Long Island at roughly the 25-meter isobed, again, out to a little bit past the shelf break, the continental shelf. These transects are represented by the black lines. And along each of these transects, we're conducting we're measuring active acoustic backscatter at 4 different frequencies, 38, 70, 120, and 200 kilohertz. Along each of these transects, we're taking hydrographic measurements, so characterizing things like the vertical profiles of temperature, salinity, fluorescence, dissolved oxygen, and other biologically meaningful parameters. Along each of these lines, we're also conducting opportunistic midwater fish trawls to characterize higher trophic organisms, like bait fish going all the way up to things like striped bass, as well as also doing zooplankton net tows at point stations, again the yellow dots, to characterize lower trophic organisms. I'm going to be talking mostly about active acoustics today, but if you'd like to learn more, you can check out Tony Ann's poster today during the poster session. In the prizes, you get to see a lot of really cute larval fish and zooplankton photos. I unfortunately won't have any more animal photos, I'm pretty sure, so I apologize for that. So back to not biology. So going into the active acoustics part of things, active acoustic surveys provide a variety of fine-scale measurements of biological aggregations. And this allows us to, when paired with hydrographic measurements and other measurements representing oceanographic processes, we are able to establish fine-scale relationships with these processes. So whether it's a change in temperature or even sea surface temperature, things like that, how that impacts fine-scale changes or fine-scale shifts, in biological distributions. So the way that we visualize and process acoustic backscatter can get very data dense and a little complicated. And so I'm going to walk everyone through what's called an echogram. This is how we primarily visualize active acoustics, and we're going to be looking at a few echograms today. So I just want to make sure that everyone is on the same page. So y-axis is depth, x-axis is our long transect distance. So that's pretty straightforward. We have hull-mounted echo sounders, so at the bottom of the ship, and those are transmitting a sound wave into the water column. And that will scatter and reflect off of various things, either in the water column or the seabed, which is represented by the darker gray region. And we are measuring those echoes. So we're getting a snapshot of where biology and other things are in the water column at any given time when we ping. And what the colors correspond to is what's known as the mean volume backscattering strength, SV, which is a super fancy way of saying the amount of stuff in a unit volume of water that reflects sound. So when we think about this biologically, you can look at kind of the reds and oranger colors representing very dense aggregations like bait fish schools and whatnot, whereas kind of these lighter blues and darker blues representing more diffuse aggregations like copepod and krill swarms. So in this echogram, again, the colored stuff, in this case, represents our biological aggregations, so biological backscatter. And then kind of the background, these kind of purplish, in some cases there'll be some white colors, those represent background or weak backscatter. It could be biological backscatter, but we can't confidently separate it due to things like background noise and whatnot. So the whole point of collecting backscatter data is to look at spatial variability, or first look at spatial variability. So again, we have those 7 transects, This represents our acoustic backscatter data over those 7 transects. Look at how things are varying spatially. And so looking at the echogram, we can just basically integrate all the backscatter in the water column to see how much biology there is over 100-meter horizontal segments. This is what's called the nautical area scattering coefficient, or NASC, which again, fancy way of saying we just add up all the stuff in the water column at any given point. This is a common proxy that's used for biomass in a variety of fisheries surveys, both Europe, Pacific Northwest, things like that. So just to walk through the plot real quick, latitude and longitude on each axis. Background color corresponds to the depth scale with 50-meter isobaths. And the brightness, so the more yellow, whiter colors, as well as larger area points correspond to higher NASC estimates. And so the echogram that we were looking at previously corresponds to just this very small segment of one part of our transect lines. So we're creating these very long echograms over 7 lines across 4 frequencies. So that's why we try to distill it into these kind of plots where you can it's more straightforward to visualize what's happening. So it turns out, unsurprisingly, that biological backscatter and aggregations are associated with boundaries. Things like bathymetry. So in the orange box here, we have the Hudson Shelf Valley, followed by the Hudson Canyon, which overlaps with over half of our transects. And it turns out that these are very biologically productive habitats because of their complex bathymetry and bottom features that can create things like local upwelling that are bringing kind of deeper, nutrient-rich water to the surface, so you get a lot of phytoplankton blooms, which then supports much more robust food webs. And you can see, looking at the echogram, it's really the only place that there's biological backscatter. And when you look at the map, there's just a giant blip there. So it's representing the same thing, just from kind of 2 different dimensions. Another important part of our survey, geographically, is the shelf break. Obviously, water column stratification doesn't only occur at the shelf break, but this is a good opportunity to discuss a little bit about the different types of stratification that we see across our survey, especially at the shelf break. And the shelf break just represents the interface between the continental shelf and the continental slope. So it goes from shallow to pretty deep very quickly. So these are echograms from the shelf break portion of each transect from 5 different surveys, but it's the same transect line. And it turns out that, similar to bathymetry, animals really like to be lazy and rest along certain isopycnals or other forms of stratification. So, for instance, during the winter, so the February and March transects shown here, the water column is pretty homogenized, it's not very stratified. Things like winter and wind-driven mixing really stirs up the water column, so you don't really see these picnic lines with these are isopycnals being displayed here with the gray lines at 0.2 kilogram per cubic meter increment. Looking at the summer and fall, you can see in the top right, the July transect, there's a very strong stratification near surface at around 25 meters and shallower. And you can see all of the biological backscatter is really pushed to the surface. It's not really below that picnic line. Comparing that to the October echogram, you can see that the picnic line is deeper and the biological backscatter is also deeper, but it still kind of rests where that picnic line is. Of course, then you also have some weird things that kind of break this mold, like when you have water mass intrusion. In this case, the Mid-Atlantic cold pool plays a pretty prominent part in the spatial distribution of marine organisms on the shelf. So here, animals are still kind of resting on specific isopycnals, but there's almost this vertical wall at the roughly 50 meter portion of the transect, which represents this very cold pool of water. That's why it's called the cold pool. So thinking about kind of the seasonal variability that you would expect in water column stratification, as well as kind of the spatial things we were talking about, we can then look at all of our surveys collectively. So each of these panels represents an individual survey at a single frequency. And to break this down, because it can be a little hard to follow otherwise, this leftmost column represents all of our spring surveys in the green box. And really the take-home point here is that NASC tends to be greater offshore. Comparatively in the summer, NASC is distributed across the shelf, so you have biomass not just near the shelf break, but both inshore, middle of the continental shelf, and at the shelf break. And you see these very large inshore blips that correspond to expected bait fish, so things like Atlantic menhaden, as well as feeding marine mammals like humpback whales that register. In the fall, you have similar distribution or broad distribution of NASC across the shelf. You don't see as many of these very strong blips from bait fish schools, but they're still present, so they're very similar in that respect. And winter, expectedly, there's just generally lower NASC. There's not as much biomass. And just to point out, our survey effort in winter is also a lot lower. Things like sea state kind of precludes conducting surveys, as frustrating as that may be. But something that's interesting is that across all seasons, across all our surveys, when we crossed the Hudson Shelf Valley, Hudson Canyon, and got out to the shelf break, when you look at everything kind of relative to each survey, that's where we see these biological hotspots from our acoustic backscatter data. So these are specific areas in our survey region that are biologically meaningful and important. So why are we using multiple frequencies? Because we look at these plots, what kind of animals are we looking at? And we can make some inferences from how backscatter varies as a function of frequency. This obviously requires significant ground truthing, so that's why we conduct our net tows and mid-water fish trawls. But there's still a fair amount of uncertainty, so just kind of keep that in mind. Generally speaking, lower frequencies like at 38 kilohertz are often associated with swim-bladdered fish or zooplankton with gas sacs, things like siphonophores. At 70 and 120 kilohertz, we're looking at small swim-bladdered fish, swim-bladderless fish like butterfish, as well as squid. At 120 and 200 kilohertz, looking at smaller fish and squid, larger zooplankton like krill and shrimp. And then at 200 kilohertz, we're really being associated with things like smaller zooplankton, copepods, chaetognaths, animals of that sort. So we went from our maps of just one frequency and we're quadrupling that. So how can we really tease out any kind of seasonal index from that information? Thankfully, a lot of really smart statisticians who do a lot of transect surveys came up with a metric for us to use, which is just calculating a stratified mean, which is shown in the red boxes in the right panel, and then 95% confidence intervals in the golden bars. So instead of looking at a giant map for each frequency across all our surveys, we have a single value and confidence interval for each survey. And so we can end up looking at plots like this where each panel corresponds to each specific frequency for the colors corresponding to winter, spring, summer, and fall from all of our surveys from 2018 to 2022. So the y-axis is our mean NASC, x-axis is our date time, and just note that there are different y-axes, and for the most part, it's very difficult to directly compare NASC values due to different resolutions of our frequency measurements. So instead, what we should really be looking at is comparing each value to the overall frequency-specific mean and 95% confidence interval, which are these dashed and dotted lines. So generally speaking, peak NASC, or we see very large values above our average NASC, occur during the summer and fall across all frequencies. So it's a common feature regardless of which frequency we're looking at. But then our middle and higher frequencies, we see relative to the overall average that winter and spring NASC can also be elevated, which may be associated with events like a spring bloom or diapause layers of copepods. So kind of the overall takeaways from this study and the work we've been doing, active acoustics can measure fine-scale distributions of biological biomass over pretty large areas. Organisms, unsurprisingly, aggregate along complex bathymetric and hydrographic features, but there are also common hotspots that seem to be present regardless of season. And that there's a strong seasonality in our biomass estimates of fish and zooplankton. So I'd just like to acknowledge the DEC for funding this work, all the ship's crew of the Seawolf and Pominock, our collaborators, and all the technicians, staff, contributors, and everyone who made this work physically possible. I certainly did not make all these measurements myself, so I thank everyone for that. And with that, I'll take any questions, Twitter handles of my co-authors as well, and happy to answer emails. Not emails. Questions now, or questions now or later.